Multiomics integration methods are increasingly needed, seeing as the volume and heterogeneity of sequencing data has increased in molecular and medical studies over the recent years. Traditionally, such methods like MOFA, for instance, rely on factor analysis. For a multi-view dataset comprising of a slew of different data modalities, such as RNA-seq, proteomics, epigenetics, and so on, such methods find shared latent factors that supposedly summarize the data, such as each factor explains one axis of variability in the data. You can think of it like PCA, where your large data with several features will be summarized by a matrix with R component, a number defined by U prior, and you can then observe each factor or component to see if they correlate with the metadata of interest in your data and could therefore be an explicative factor for your data. However, these metadata are almost always analyzed a posteriori, as you can see. However, if you have, for instance, a pharmacogenetic multi-view dataset where each of your sample has been treated with a particular drug before sequencing, then for each combination of sample and feature, you have much more measurements, as many as you have drugs. Conceptually, instead of representing the data in a flat matrix manner, you could represent it in a 3D object called a tensor, where each value represents a unique omic measurement for a patient treated for a particular drug. The question then is, how can we exploit this representation? Now that we have baked the metadata inside the input, how do we use it to strengthen the explanative power of our model? There are methods called tensor decomposition methods to analyze such objects. And that's from where we went to develop MUSIC, standing for Multi-View Bayesian Tensor Decomposition. Given a collection of 2D or 3D datasets showing common samples, MUSIC is capable of decomposing the dataset into a set of 3 shared latent factors called factor matrices that explain most of the variability in the data. Each factor matrix summarizes variance along one dimension of your data, such as the samples, the features, or another aspect that constitutes a third slide of your tensor, such as drug treatment or cell type. These factor matrices can reveal insight regarding one of those aspects of the data, similar to PCA, but they can also be combined together through matrix multiplication to create what we call loadings, that are combinations of 2 factor matrices in a latent space into a latent tensor. For instance, you can combine the features and the cell type's factor matrices into a cell type times features times number of factor tensors. It is the strength of MUSIC, as it can allow you to analyze in the latent space 2 aspects of the data in regard to each other, which traditional factor analysis method can do. We coded MUSIC with a Bayesian framework, which allows us to introduce structured sparsity prior, as can be seen in the plate model. That plate model shows that your inferred tensor, here denoted as Y, has as priors the 3 normally distributed matrices on which we impose horseshoe priors for regularization. And this allows us to model different aspects of variability of the data. For instance, gamma here models noise, while delta models the variability of the features specifically in an attempt to model innate biological variance. MUSIC has been shown to give very satisfactory results when it comes to data reconstruction, performing similar to TensorLee's implementation of Parafac and being more stable as the number of samples increases. When it comes to real data, however, we had 2 main questions. First, can MUSIC preserve the biological integrity of the dataset? And second, can it discover novel insights in datasets? For the first one, we used the dataset of CLL cell line stimulated with various cytokines and drugs and whose survival was assessed. Additionally, RNA-seq analysis was performed for this dataset. We then ran MUSIC twice, once in a single view setting with only the survival values, and a second time adding the RNA-seq as a way to prove the importance of jointly modeling omic modalities alongside the rest of the data. Indeed, we show that, as such, we can obtain a better separation of the drug factors when it comes to whether or not they impact the BCR pathway. Now, about novelty, to that end, we used single-cell RNA-seq and single-cell ATAC-seq of ALL pediatric patients and healthy controls. We transformed the data into a pseudo-bulk dataset by averaging normalized values to the patients as well as the cell type level, giving for each omic view a patient times cell type times features tensor. We used the cell type annotation of the study. While MUSIC was perfectly capable of finding individual factors that separate the ALL and healthy patient, the patient factors revealed a subsetting of ALL patients into two subgroups, which was mostly driven by factor 0. We then decided to inspect that factor further, and to that end, we looked at the cell type times gene loadings, obtained through matrix multiplication of the gene and cell type factor matrices, and we zoomed in on the factor value in factor 0. Like I said earlier, MUSIC is unique in that it allows this representation of factor values for features in regard to the cell types in the latent space. With that, we can clearly see a distinct PDC signature for this factor, and what is interesting is that this signature exhibits up and down regulated gene as well as upregulated pathways that are not common in B-cell driven ALL and actually more common in general in T-cell driven ALL. This difference could be an explanation as to the existence of this subgroup of patients. So in conclusion, MUSIC is a probabilistic model that allows the decomposition of higher order tensor and therefore the analysis of several axes of variation jointly in the latent space, which is not possible by more classical multi-omic integration method, while naturally extending this to a multi-view setting, which traditional tensor decomposition methods like Parafac cannot do. Thank you for your attention.